So today we are going to discuss about one of the most energy-rich organic macronutrient, that is lipid. Lipids are broad group of naturally occurring molecules that include fats, waxage, sterols, fat-soluble vitamins, monoacylglycerols, diacylglycerols, phospholipids, and others. Lipids are those organic compounds which are generally soluble in organic solvents and largely insoluble in water. Main biological function of lipids include energy storage. As you know, lipid provides 9 kilocalorie per gram of lipids. It also plays important role in the structural component of cell membranes and also function as fat-soluble vitamins, specifically vitamin A, D, E, and K. This has also important biological function as hormones and important signaling molecules. Lipid is sometimes used as a synonym of fats in general day-to-day language, but there is a great difference as fats are a subgroup of lipids called triglyceride or triacylglycerols, whereas lipid encompass molecules such as fatty acids and their derivatives, including tri, di, and monoacylglycerols and phospholipids, as well as other sterol-containing metabolites such as cholesterol. Humans use various biosynthetic pathways for both degradation and synthesization of lipids. Some essential lipids cannot be made this way, so they must be obtained from the diet on regular basis, otherwise they will lead to deficiency disorders. Lipids are broadly classified into 7 groups, including fatty acids, acylglycerols, glycerophospholipids, sphingolipids, sterols, prenol-lipids, and saccharolipids. We will discuss each individually now. First comes fatty acids. You know, this fatty acid consists of hydrocarbon long chain and a terminal carboxylic acid group. This arrangement confers the molecule with a polar hydrophilic end that is made up of carboxylic acid group and a non-polar hydrophobic end that is made up of that hydrocarbon chain, which is obviously insoluble in water, being hydrophobic in nature. Fatty acid structures are the most fundamental categories of biological lipids, which is used as a building block for more structurally complex lipids. This fatty acid carbon chain containing from 4 to 24 carbon atoms in the same chain may be saturated or unsaturated. Saturated fatty acids contain all saturated carbons with hydrogen atoms, and its general formula is CH3, CH2, N, COOH, where N is an even number. Unsaturated fatty acid may be monounsaturated fatty acid or polyunsaturated fatty acids. Monounsaturated fatty acid will obviously contain only one double bond in their structure, whereas polyunsaturated fatty acids will contain 2 or more double bonds in their structure. Double bonds in polyunsaturated fatty acids are generally separated by at least one methylene group. In case of double band, adjacent hydrogen molecules will provide its geometric isomerism configuration. That is, this structure may be either cis or trans geometric isomerism, which will significantly affects molecules molecular configuration. It has been observed that in case of cis double bonds, where both the hydrogen atoms adjacent to the double bond are on the same side, that cause the fatty acid chain to bend on other side comfortably, which is an effect that is more pronounced when more double bonds are there in a chain. This cis double bond plays important role in structure and function of cell membranes. Naturally occurring fatty acids are having more of cis configuration, although transforms are also seen in natural and partially hydrogenated fats and oils. You should remember one important thing, that shorter is the chain, lower will be its melting point, and even fatty acid chain containing more of double bonds, that is unsaturated fatty acids, are having lower melting temperatures as compared to saturated fatty acids of the same length. Now comes second second type of lipid, that is glycerolipids. Glycerolipids are composed mainly of mono, di, and tri-substituted glycerol. The most well-known is triacyl glycerols, which is also known as triglycerides or fats. In these compounds, all the three hydroxyl groups of glycerol are, here you can see, all the three hydroxyl groups of glycerol are esterified with different fatty acids, resulting in mixed lipids. This is structure of triacyl glycerol. This has important function as a food store. These lipids comprise bulk of storage fat in animal tissues as well as in oil seeds. Triglycerides or fats may be solid or liquid at room temperature, depending upon their structure and composition. If it will contain more of unsaturated fatty acids, obviously it will be liquid at room temperature, but if it will contain more of saturated fatty acids, it will result in solid formation at room temperature. There are different terms for it. Oil is the term used to refer to fats which are liquids at normal room temperature having more of polyunsaturated fatty acids, whereas the term fats are used to refer to fats which are solid at normal room temperature and contain more of saturated fatty acid in that. Lipids term is used to refer both liquid as well as solid fats along with other related substances. Now comes next group, that is glycerophospholipids. These glycerophospholipids are also known as phospholipids, and these phospholipids are key components of the lipid bilayers of cells. Glycerophospholipids are also involved in metabolism and cell signaling. Neural tissues, including brain, contains relatively high amounts of this glycerophospholipids, and even slight alteration in their composition has been implicated in various neurological disorders. Phospholipids are found in biological membranes in the form of phosphatidylcholine, phosphatidylethanol and phosphatidylserine. Even plasmalogens are there, which is a type of glycerolipids which contain fatty alcohol at C1 of SN glycerol with double bond instead of a fatty acid. Here you can say simple structure of glycerophospholipids, which is having two molecules of fatty acids attached to glycerol backbone, and one molecule of phosphate ester that is polar in nature. Polar means that is hydrophilic in nature, and these fatty acids make apolar nature, that is hydrophobic in nature. Here you can see simple structure of different glycerophospholipids. This is simple structure of L-glycerol 3-phosphate, where phosphate group is attached to C3 of the normal glycerol. These glycerophospholipids are basically amphiphilic in nature. You have seen because same molecule contains both hydrophilic as well as hydrophobic parts. And here, this X is attached to phosphate group, which is representing the molecule attached to that phosphate group, and this molecule will be responsible for the nature of glycerophospholipids, which is formed by that particular molecule, which is attached here. As for example, you can see if this X is replaced by H, it will form phosphatidic acid. Whereas if ethanol amine replaces this X, there will be formation of phosphatidyl ethanol amine. If this X is substituted by choline molecule, this will form phosphatidyl choline. Similarly, serine moiety attached to that phosphate group will lead to the formation of phosphatidyl serine, and if this this X is replaced by glycerol, it will form phosphatidyl glycerol molecule. If this X is replaced by the molecule, the moiety myoinositol 4,5 bisphosphate, there will be formation of phosphatidyl inositol 4,5 bisphosphate glycerophospholipid. And if this X is replaced by phosphatidyl glycerol, there will be formation of cardiolipin. So these are different glycerophospholipids depending upon the moiety attached to that phosphate group in the basic structure of glycerophospholipids. Next comes sphingolipids. Sphingolipids are complex family of compounds that share common structural feature with a sphingoid base backbone, which is synthesized de novo from the amino acid serine and a long chain fatty acid OA. And this is then converted into either ceramides, phosphosphingolipids, glycosphingolipids, or in other compounds of sphingolipids. Major sphingoid base of mammals is commonly referred to as sphingosine. Ceramides and acyl sphingoid base are a major subclass of sphingoid base derivatives with an amide linked fatty acids. The fatty acids are typically saturated or monounsaturated with chain lengths from 16 to 26 carbon atoms. This is basic structure of sphingolipid, and this is backbone of that sphingolipid, that is sphingosine. Here you can see in sphingolipids, the hydrophobic region consists of a long chain sphingoid base with generally 18 carbons such as sphingosine, which is linked to the acyl group of a fatty acid via an amide bond R2. The hydrophilic region R1 consists in the simplest case of a hydroxyl group in the case of ceramide, which we will see in next slide. Here you can see the gender structure of sphingolipid. There is sphingosine backbone, there is fatty acid, and here with C3 in place of X, whatever moiety will be again attached, this will represent the type of sphingolipids. In case of ceramide, that X is replaced by hydrogen bond, whereas if it is replaced by phosphocholine, there will be formation of sphingomyelin. If this X is replaced by glucose moiety, there will be formation of neutral glycolipids, glycosyl cerebroside. If it is replaced by di, tri, or tetrasaccharide, there will be formation of lactosyl ceramide. And if this X is replaced by complex oligosaccharides, there will be formation of ganglioside GM2. So these are different types of sphingolipids and their structures. Now let us see functions of mammalian sphingolipids. Sphingolipids are found to protect the cell surface against harmful environmental factors by forming a mechanically stable and chemically resistant outer leaflet of the plasma membrane lipid bilayer. Certain complex glycosphingolipids are involved in specific functions of cell recognition and signaling. Cell recognition depends on physical properties of sphingolipids, whereas signaling involves specific interactions of the glycan structure of glycosphingolipids with similar lipids present on neighboring cells or with proteins. Next comes sterols. Sterol lipids, for example, cholesterol, which is an important example of this group, and its derivatives are important component of membrane lipids, along with the glycerophospholipids and sphingomyelins. Steroids are derived from the same fused four-ring core structure that have different biological roles as hormones and signaling molecules. 18 carbon steroids include the estrogen family, whereas 19 carbon steroids, that is C19, comprise the androgens such as testosterone and androsterone. Whereas C21 subclass comprises of progesterones as well as glucocorticoids and mineralocorticoids. Corticosteroids comprising various forms of vitamin D are characterized by cleavage of the B ring of the core structure. Other examples of sterol includes bile salt and their conjugates, which in mammals are oxidized derivatives of cholesterol and are synthesized in the liver. This is basic structure of cholesterol, which is a steroid in which one of the hydrogen atom is substituted with a hydroxyl group in the carbon chain. They have in common with steroids the same fused four-ring core structure. Next comes prenol lipids. Prenol is a natural alcohol and it is one of the simplest terpenes. Prenol lipids are synthesized from 5 carbon precursors isopentenyl diphosphate and dimethylallyl diphosphate, which is produced mainly via mevalonic acid pathway. Simple isoprenoids, that is linear alcohol, diphosphates, etc., are formed by successive addition of C5 units and are classified according to number of these terpene units. Structures containing more than 40 carbons are known as polyterpenes. Carotenoids are important simple isoprenoids which function as antioxidants and as precursor of vitamin A. Another biologically important class of molecules is exemplified by the quinones and hydroquinones, which contain an isoprenoid tail attached to a quinonoid core of non-isoprenoid origin. Vitamin E, vitamin K, and ubiquinones are examples of this class. Next comes sacharolipids. These are compounds in which fatty acids are directly linked to sugar backbone, forming structures which are compatible with membrane bilayers. In sacharolipids, a monosaccharide substitutes for the glycerol backbone present in glycerolipids and glycerophospholipids. Most familiar sacharolipids, that is acylated glucosamine precursors of the lipid A component of the lipopolysaccharides, are available in gram-negative bacteria. Now, in this table, you can see different sources of common fatty acids. Here, butter fat contains most of the butyric acid and caproic acid, whereas coconut oil provides lots of caprylic acid, capric acid, and lauric acid. Palm kernel oil and palm oil supplies good amount of myristic acid and palmitic acid respectively, whereas animal fats are good source of palmitoleic acid and stearic acid. Olive oil contains lots of oleic acid, and butter fat supplies lots of vaccinic acid. Grapeseed oil provides good amount of essential fatty acid, that is linoleic acid. Flaxseed oil supplies alpha linolenic acid, that is also essential fatty acids. Borage oil provides lots of gamma linolenic acid, whereas fish oil and peanut oil are good source of arachidic acid. Liver fat supplies good amount of arachidonic acid, whereas fish oil are good source of eicosapentaenoic acid. That's all about lipids. Thank you. Thank you very much.
Thank you, ma'am.
Thank you, ma'am.
Good evening, sir.